Russel, I was wanting to know, you showed the males that had not a lot of dendritic extensions, that beautiful slide you showed on the non-dominant male versus the dominant. Is the female similar to the non-dominant?
So this is a great question. Actually, what I showed you was the change between reproductively inactive and active. And they grow those cells much larger, 8 times larger, in fact. Both the males and the females do. The males are driven by their social situation. The females follow an intrinsic cycle. So when they become reproductively active, they will also enlarge their cells. They'll wire them together, and we don't know that yet, but we suspect they wire them together, and then they become able to produce pulsatile GnRH, which is necessary for reproduction. So that it's 2 different systems that are doing the same thing, but under different control.
So the brakes on the pulsatility of the GnRH for the male makes him, the subordinate male, the brakes are on.
Right.
Then the female, the brakes come off the GnRH and they become pulsatile when what?
When she's gravid and ready to lay eggs. Art.
I had a question for Marius Wernig. When you make an iPS cell re-express the inactive X chromosome, has anybody studied the transcriptome to see what changes relative to a very similar cell that has one X chromosome inactive?
Yes, people have looked also using
Globally.
microarrays, global RNA sequencing, and by all means, this is an active X chromosome, and it's so yeah.
And how much does it influence the autosomal? I mean, having 2 active X chromosomes later in life is lethal. So why is it that these cells can tolerate having 2 active X chromosomes?
That's a fantastic question, and I guess nobody really knows the answer. There's a couple of very unique features about these pluripotent cells that no other somatic cells can tolerate. For example, we touched upon this DNA methylation. Same thing. When you lose global DNA methylation in any somatic cells, these cells will die from apoptosis very rapidly. A very different story in these pluripotent cells. You can strip them of all the DNA methylation and they're still perfectly fine. They cannot differentiate anymore because as soon as they differentiate, they will die. But as stem cells, they're perfectly fine. So there's a couple of very interesting features that we don't really understand that they can deal with.
Is it the DNA methylation of specific genes or the?
Global. You can knock out all 3 DNA methyltransferases. You can detect any CpG methylation anymore, and they don't seem to mind.